Everything you have been told about fasting and muscle loss is built on a misunderstanding of what autophagy actually does inside a muscle cell. The fear is specific and widespread. Extended fasting activates autophagy. Autophagy breaks down protein. Muscle is protein, therefore fasting eats your muscle. The logic chain seems airtight. It is also wrong at the third step. Research published in Cell Metabolism in 2009 tested what happens to skeletal muscle when autophagy is blocked entirely, when scientists deleted the gene ACT7, which is essential for autophagy, specifically in muscle tissue. If the fear were correct, muscle without autophagy should be protected. It should be preserved. It should retain mass and function better than muscle with autophagy running. The opposite happened. Muscles lacking the capacity for autophagy developed profound atrophy. Force decreased with age. The muscles accumulated abnormal mitochondria, damaged sarcoplasmic reticulum, and disorganized sarcomere structure. And when these autophagy-deficient muscles were subjected to fasting, the muscle loss was worse than in normal muscle, not better. The researchers concluded directly, autophagy flux is important to preserve muscle mass and to maintain myofiber integrity. Autophagy is not the mechanism that destroys muscle during fasting. It is the mechanism that keeps it intact. What actually happens to your muscles when autophagy peaks is the opposite of what most people fear, and the timeline of how it unfolds changes how you should think about fasting entirely. Think of your muscle tissue as a building that has been continuously occupied and in use for decades without a full renovation. The building is still standing, the load-bearing walls are intact, the foundation is solid, but inside every room there is accumulated damage, fraying electrical wiring that is still functional but running less efficiently than it should, cracked plumbing that is still delivering water but at reduced pressure, structural elements that were stressed and micro-damaged but not replaced because the building never closed long enough for the renovation crew to access them. Autophagy is not demolition, it is selective renovation. It targets the fraying wiring, the cracked pipes, the damaged internal components, specifically and precisely, without touching the load-bearing walls, the foundation, or the structure of the building itself. When autophagy peaks in skeletal muscle, it is renovating the interior of every room. The building does not weaken. It emerges with a cleaner, more functional interior than it had before the crew arrived. Here is what the timeline actually looks like. Hours 0 to 16, the fear zone. This is the window where most concern about muscle loss during fasting is focused, and it is also the window where the least muscle-specific change from autophagy is occurring. During the first 16 hours without food, the body is drawing down glycogen reserves and beginning to shift fuel sources. Insulin is dropping, AMPK is rising. The conditions for autophagy are building, but in skeletal muscle specifically, the autophagy markers LC3 flux and P62 degradation have not yet shown measurable significant elevation in human studies. What is happening to muscle protein in this window? Research by Cahill and colleagues, referenced extensively in modern fasting literature, mapped the sequential fuel hierarchy during fasting and confirmed that significant mobilization of muscle protein is a last resort, not a first response. In the first 16 hours, the body is running primarily on stored glycogen and beginning to shift toward fat oxidation. Muscle protein catabolism for energy is minimal. The fear of this window is larger than the biological reality. Hours 16 to 24, autophagy begins in muscle. Research measuring autophagy markers in human and animal skeletal muscle finds that LC3 and P62 begin shifting measurably around the 16 to 18-hour mark of fasting. The autophagy system in muscle is activating. But what is it targeting? This is the critical distinction that the fear-based framing always misses. Inside a muscle cell, there are two categories of protein: contractile proteins, the actin and myosin filaments that actually generate force, and everything else. The cellular housekeeping proteins, the damaged mitochondria that have been accumulating oxidative damage from repeated high-intensity contractions, the misfolded structural proteins from exercise-induced stress that were not fully cleared during previous recovery periods, the aggregated metabolic byproducts that have built up in the cell over months of training. Autophagy selectively targets the second category, not because it is programmed to ignore contractile fibers, but because damaged and misfolded proteins are the substrates that the autophagy machinery is specifically designed to identify and process. Intact, functional contractile proteins do not carry the molecular tags that mark damaged cellular material for autophagy. A study published in the Journal of Applied Physiology by Dethlefsen and colleagues in 2017 measured skeletal muscle autophagy at 2, 12, 24, and 36 hours of fasting in human subjects. The finding confirmed that in untrained individuals, autophagy markers in skeletal muscle showed measurable changes. And crucially, the changes were consistent with clearance of damaged cellular content rather than breakdown of structural muscle. The renovation crew is in the building. They are pulling out the damaged wiring. The load-bearing walls are not touched. Hours 24 to 36, the mitochondrial renovation. This is the window where one of the most consequential aspects of muscle autophagy operates, a specialized subtype called mitophagy, the selective autophagy of damaged mitochondria. Skeletal muscle has one of the highest mitochondrial densities of any tissue in the body. Every contraction, every training session, every bout of sustained effort generates reactive oxygen species inside mitochondria, oxidative damage that accumulates in the mitochondrial membrane and electron transport chain over time. A mitochondrion that has accumulated sufficient damage becomes less efficient. It produces less ATP per unit of oxygen consumed, generates more heat, and contributes more oxidative stress to the surrounding cell. These dysfunctional mitochondria do not simply stop working. They continue running at reduced efficiency, producing more free radicals, competing for the cellular resources that healthy mitochondria need. Mitophagy removes them selectively. The 24 to 36-hour autophagy window is the period in which mitophagy operates at a depth that shorter fasting windows cannot reach, processing the backlog of damaged mitochondria that has accumulated from repeated training cycles. Research on athletic performance consistently finds that the quality of mitochondria, not just the quantity, determines aerobic capacity and endurance performance. An athlete whose muscle tissue contains a higher proportion of high-functioning mitochondria produces more force and recovers faster than an athlete with the same mitochondrial density but more accumulated mitochondrial damage. The renovation crew, in hours 24 to 36, is specifically renovating the power generation units of every room in the building, not removing rooms, replacing the old generators with the equipment that runs cleaner. The growth hormone factor. Running in parallel to the autophagy timeline is a hormonal dynamic that directly addresses the muscle protein question. As fasting extends through the 24 to 48-hour window, growth hormone surges. Multiple studies have documented increases in growth hormone during extended fasting. Growth hormone is a counterregulatory hormone. It signals the body to preferentially oxidize stored fat rather than catabolize muscle protein for energy. It is one of the primary evolutionary adaptations to periods without food: preserve functional tissue, draw down energy reserves. In practical terms, this means that as autophagy in muscle is running at peak depth in the 24 to 36-hour window, growth hormone is simultaneously running at elevated levels, specifically to protect muscle from the protein catabolism that the fear-based framing assumes is occurring. The building's security system is active precisely when the renovation crew is doing its deepest work. What about the protein release? The PLOS-1 study published in 2014 measuring the effects of a 72-hour fast on human skeletal muscle protein metabolism found that fasting did significantly increase net phenylalanine release from forearm muscle, an indicator of net protein loss from muscle. The mTOR pathway was suppressed by approximately 50%. This finding is real and worth understanding accurately. During extended fasting, some muscle protein does turn over. The question is which proteins are turning over and why. The autophagy research provides the context. The proteins turning over are preferentially the damaged, misfolded, and aggregated proteins that have accumulated in the cell and that autophagy has been processing. These are not contractile proteins contributing to muscle force production. They are the cellular debris that has been reducing efficiency. Additionally, when autophagy processes a damaged protein, the resulting amino acids are not lost. They are released into the intracellular amino acid pool and can be used for immediate synthesis of new, functional proteins. Autophagy is not simply degradation, it is recycling. The renovation crew is not throwing materials into a disposal site. They are sorting, reclaiming usable materials, and sending them to where new construction is needed. The refeeding window. What happens to muscle after autophagy peaks is determined almost entirely by the first meal after the fast. Muscle protein synthesis is suppressed during extended fasting because mTOR is suppressed. When food, specifically protein, arrives after the fast, mTOR reactivates rapidly, and the cellular machinery for muscle protein synthesis has a clean, cleared cellular environment in which to operate. Research on time-restricted eating and protein intake timing consistently shows that the anabolic response to protein consumed after an extended fasting period can be robust, not blunted. A 5-day fasting mimicking diet study published in the European Journal of Applied Physiology in 2021 found that participants preserved muscle function and strength throughout the protocol, with no reduction in muscle fiber size observed in biopsies. The renovation creates the conditions for better construction afterward. The cleared mitochondria, the removed damaged proteins, the cleaner cellular environment, all of it makes the rebuilding response to the post-fast meal more efficient than it would be if the cell were still carrying the debris load. Before the protocol, this content describes biological mechanisms in healthy adults. Extended fasting to reach autophagy peak in skeletal muscle is not appropriate without prior experience with shorter fasting windows. If you have diabetes, kidney or liver disease, a history of disordered eating, or take ongoing medication, speak with your doctor before attempting extended fasting protocols. Electrolyte management during fasting is critical. Nothing here treats or diagnoses any medical condition. The practical summary is this: the fear that autophagy peaks your muscles away is built on a misread of which proteins autophagy targets. The research that tested what happens when autophagy is blocked in muscle found the opposite of protection. The renovation crew that runs at peak in the 24 to 36-hour window is the same crew that keeps the building standing long-term, and the post-fast meal builds on a foundation that the renovation has improved. If this changed how you think about what is actually happening inside your muscle during extended fasting, subscribe and leave a like. Tell me in the comments, did you avoid fasting because of muscle loss fear, and did this change how you see the mechanism?